How can a fully formed organism with complex organs and tissues, like the heart, brain, muscles, and bones, come from a single fertilized egg cell? In this video, we'll explore stem cells and differentiation, which play crucial roles in the development of complex organisms. First, we'll look at cell specialization with some key examples of specialized cells' form and function. We'll also look at limits to cell sizes, including the surface area to volume ratio. We'll discuss stem cells and some of their unique capabilities, and we'll also consider stem cell niches, or tissues that contain stem cells in fully formed organisms. These are some of the most important topics covered in the IB biology syllabus. When we consider the structures of plant cells or animal cells, we're usually looking at a generalized cell. A generalized cell has all of the necessary organelles to sustain life. However, as cells specialize, they may lose some of these organelles and begin to rely on other cells in the organism to provide for those needs. For instance, erythrocytes, or red blood cells, do not have a nucleus and therefore cannot reproduce. Red blood cells depend on blood stem cells to carry out reproduction to create more red blood cells. Specialized cells will have a structure that allows them to perform their functions efficiently. For instance, neurons are specialized to transmit electrical signals through their long, thin axons, which are insulated by a myelin sheath. Female gametes, or eggs, contain all the organelles needed for rapid cell division after fertilization, making them large and rich in cytoplasm and organelles to support the early stages of embryonic development. In contrast, male gametes, or sperm cells, are adapted to compete in reaching the egg first to deliver their DNA. They are streamlined and lack excess organelles that could slow their speed. Instead, they have flagella powered by numerous mitochondria to enable rapid movement. Red blood cells, or erythrocytes, do not have a nucleus, allowing them to have a flat and inverted cell surface that increases their surface area for releasing and absorbing oxygen across their membrane. Striated muscle cells are long and lean and have actin and myosin proteins to allow for contraction, and they contain many nuclei to support their longer structure. As we have seen in our examples of specialized cells, cells can come in a variety of sizes that allow them to perform their functions well. However, regardless of the size of the cell, they are limited in their size by their ability to exchange nutrients and waste across the membrane. As the cell grows larger, the volume of the cytoplasm contained within it increases. The increase in cytoplasm also means there'll be more organelles present, and that increases the need to exchange nutrients like oxygen, as well as wastes. A larger cell also increases the distance for molecules like oxygen to diffuse in, as well as for waste to diffuse out. As the cell grows, the cell membrane expands, but not as rapidly as the cytoplasm. As the cell increases in size, the volume of the cytoplasm increases fourfold, while the surface area of the plasma membrane only doubles. Eventually, the volume of the cytoplasm becomes too large for the surface area of the membrane to keep up with the exchange of nutrients. Let's see why this is using cubes as a model for cells. Although most cells aren't in this cubic shape, the mathematical growth model works the same for different cells, and it's simpler for us to use the block shape. This small cube here has a length of 1 centimeter. The surface area just measures the outside surface of the cube, so we'll measure the area of one square using length times width, then times it by the number of sides, and that gives us a surface area of 6 centimeters squared. Notice that we're only measuring the surface here, so we're measuring in just 2 dimensions, length and width, giving us the centimeter squared unit. Now, to measure volume, we'll use the formula for volume, length times width times height, and it has a volume of 1 centimeters cubed. Notice we're using the centimeter cubed as we're measuring in 3 dimensions, length, width, and height. So we have a 6:1 surface area to volume ratio for this small cube representing a small cell. We can imagine that for every unit of cytoplasm volume, there are 6 units of membrane surface area to support exchanges of nutrients and waste. Let's see what happens when the cube doubles in length. Now the cube has a surface area of 24 centimeters squared and a volume of 8 centimeters cubed. Giving us a 24:8 surface area to volume ratio, we can reduce down to 3:1. Now for every unit of cytoplasm, we have just 3 units of the membrane surface area to support it. And if we double the length again, now we have a surface area to volume ratio of 2. Now for every unit of cytoplasm, we have just 2 units of membrane to support it. This low surface area to volume ratio indicates a decreased capacity to exchange materials across the membrane to support the cell's needs. I like to think of the surface area to volume ratio as a measurement of the cell's efficiency in exchanging materials. As the cell size increases, it becomes less efficient, and the surface area to volume ratio decreases. When a cell grows too large to exchange materials efficiently, it must divide in order to increase its surface area to volume ratio. If a cell is limited in size, how can a neuron reach lengths of a meter or more? Cells can be of various sizes as long as the surface area to volume ratio is efficient enough for the exchanges it requires. Although the neuron can become quite long, the cytoplasm is stretched in the narrow structure of the axon. This increases the surface area for exchange. So although the neuron might be a meter long, its surface area to volume ratio is relatively high. We find that cells that are involved in exchange as part of their function typically have high surface area to volume ratios. One example of a cell that has evolved a high surface area to volume ratio because of its function to exchange materials is a proximal convoluted tubule cell. This example from the higher level content is a cell whose function it is to selectively reabsorb nutrients and solutes from the filtrate in the kidney. They have microvilli that form a brush border. This greatly increases the surface area of the membrane, which allows for more materials to be transported into the cell during selective reabsorption. The base of the cell includes invaginations of the membrane, which also increases the surface area for nutrients to move back into the bloodstream through the capillary. Type 1 pneumocytes can be found in the alveoli of the lungs. Oxygen must diffuse across these cells to enter the capillary for absorption into the red blood cell. The type 1 pneumocyte has a flattened shape, making it very thin. This shortens the distance needed for oxygen to diffuse, which increases the rate of diffusion as they move from the alveolus into the capillary and red blood cell. Type 2 pneumocytes' main function is to produce pulmonary surfactant, or amphipathic fluid that consists of phospholipids. This keeps the moist surface of the alveoli from sticking together due to water's adhesive and cohesive properties. Type 2 pneumocytes are cuboidal or have a cube-like shape, which allows for more organelles that are involved in surfactant production, including vesicles for export. They also have microvilli that are facing the lumen of the alveoli, which allows for more surface area to secrete surfactant. Meanwhile, red blood cells, or erythrocytes, have a concave center that increases the surface area. The curvature also limits volume, driving the surface area to volume ratio up and allowing it to exchange oxygen efficiently. Red blood cells do not have a nucleus, which allows a hollowed-out shape, and so they must be renewed by hematopoietic stem cells, or HSCs. Stem cells are generalized cells that are responsible for creating specialized cells through the process of differentiation. Recall that in sexually reproducing organisms, the zygote is the first cell of a new organism that occurs when a male gamete fertilizes a female gamete. The zygote is an unspecialized stem cell capable of becoming any cell in the embryo or placenta. The zygote will undergo multiple rounds of divisions, creating tissues that will begin to specialize through the process of differentiation. All cells in the tissues have the exact same genetic information, copied from the original zygote, and therefore the genetic capacity to become any type of cell. Cells will begin to specialize by suppressing or expressing different patterns of genes. Striated muscle cells will have similar patterns of gene expression compared to other muscle cells, but very different patterns of gene expression compared to neurons. Gene expression is the transcription of a segment of DNA that will be translated into a polypeptide to build a protein. These patterns of gene expressions are controlled by different chemical signals, as well as gradients in chemical signals as the embryo develops. As the tissues start to specialize, they start to limit their capacity to become any type of cell. Therefore, the early embryo has the most potent stem cells capable of becoming a wide range of cells. As the embryo develops and cells begin to specialize, the cells lose their ability to become different types of cells. We can classify the ability of stem cells to become different cells using the following terms. Totipotent stem cells are the most powerful stem cells, as they can become any type of cell in the embryo or placenta. Pluripotent stem cells can develop into any cell in the embryo. Multipotent stem cells can develop into a narrow range of different types of cells. For instance, blood stem cells that can differentiate into white blood cells, red blood cells, or platelets. Finally, we have unipotent stem cells that can only produce one type of cell but can do it indefinitely. For instance, germ-line stem cells that produce sperm cells. You can use the mnemonic top purple mice at uni to remember the order of potency for stem cells. To understand how these different potencies of cells apply to development, let's take a look again at the stages of development. The zygote is the first cell formed from the fertilization of the egg cell from a sperm cell. The zygote is a totipotent stem cell, as it can differentiate into any cell found in the embryo or placenta. It is the most powerful of stem cells, as it is the most versatile. Shortly after fertilization, the zygote divides rapidly, forming the blastocyst. Within the inner layer of the blastocyst, we find pluripotent stem cells. These stem cells are not capable of becoming cells in the placenta, so their ability to specialize is already a bit limited. However, they are very powerful stem cells, as they can become any cell within the embryo, and they will give rise to the first stages of differentiated tissue of the ectoderm, mesoderm, and endoderm. These cells are often harvested for research as they have a wide scope of versatility. As the embryo develops, its pluripotent stem cells differentiate into specialized tissue and lose their capacity to become any cell in the body. However, when a baby is born, and even as adults, the human body contains adult stem cells. Many of these stem cells are multipotent, meaning as they divide, they either remain unspecialized to renew the pool of multipotent stem cells, or some will differentiate into a narrow range of cell types. Some adult stem cells are unipotent, meaning they can remain undifferentiated to renew themselves, or they differentiate into only one cell type. Stem cells will receive chemical signals to either remain undifferentiated or different chemical signals that prompt them to specialize. Therefore, multipotent and unipotent adult stem cells are found in specific areas of the body that have highly specialized environments known as stem cell niches. A stem cell niche is a very specific chemical environment that regulates stem cell growth and regeneration. Adults have multipotent and unipotent stem cells contained in stem cell niches. This encourages some stem cells to not differentiate in order to regenerate the stem cell pool, while also encouraging some stem cells to differentiate into specialized cells to regenerate tissues. There are several stem cell niches in the body, including bone marrow, where hematopoietic stem cells, or HSCs, can differentiate to form the different components of blood, including red blood cells, white blood cells, and platelets. HSCs are considered multipotent as they can differentiate into more than one cell type. Another stem cell niche is found near hair follicles that regenerate the hair follicle that produces hair. In this video, we saw how specialized cells have a structure that allows them to perform their function efficiently. We also saw that cells are limited in size by their surface area to volume ratio, or their efficiency in exchanging materials across their membrane. We reviewed how some cells can be quite large, but they will still have a high enough surface area to volume ratio for efficient exchange, like the neuron. We also reviewed how stem cells can differentiate into different cells in the body and that the early embryo has the most potent stem cells. We looked at stem cell niches, which are microenvironments in the body that contain multipotent or unipotent stem cells that can renew themselves or differentiate into a limited number of cells.